Hello, everybody. Welcome back to our session or Series A on vertebrate genomes. And I hope everybody's enjoying the conference. I think it's been a good success so far. And in this session, we're going to cover some highlights of research that's ongoing with vertebrate genomes. And in this case, that includes projects that are part of the Vertebrate Genomes projects, but others like the Bird 10K project, the Cetacean Genomes project, and so on. And so whether or not these groups are using long reads, short reads, or whatever approach, we want to hear what's going on out there in the vertebrate world. As usual, we're going to hear from 5 speakers today, and we're going to let them have their videos show up. And I encourage you to put questions into the chat window. We'll be screening those questions and ask them to the speakers. And then after each talk, we'll have a question and answer session. So first up is Josephine Stilner from the University of Denmark. She'll be talking about the Bird 10K Genomes project. She's a postdoc with Goji Zhang. And so Josephine, take it away.
Hello from Copenhagen. My name is Josephine Stille, and I'm a postdoc in Goji Zhang's lab at the Willems Center for Biodiversity Genomics at the University of Copenhagen. Today, I will give you a brief overview of the 10,000 Bird Genome Project, a project that aims to generate draft genomes for each of the living bird species. And I will give you a brief overview of its history, its current status, and where we're going in the future. Why would we want to sequence genomes for all bird species? As I'm showing here on the right-hand side with these beautiful paintings by Johan Fieldsø, birds have a tremendous variety of phenotypes. They're ecologically, physiologically, and morphologically diverse. They're distributed worldwide. They swim, they run, they dive, and of course they fly. And birds have also been relatively well studied compared to a lot of other groups. So we have good baseline knowledge that allows us to integrate the trait data with the genomic patterns we see. We also keep birds as pets and we eat them. And a number of bird species are important biomedical and evolutionary models. The bird phylogeny is possibly one of the hardest phylogenetic problems, and this is where my main interest with the B10K data lies. There are long-standing questions regarding the branching order of the deep nodes of the bird phylogeny, and this is regardless of large amounts of data that have been used to try to resolve those patterns. From a practical point of view, bird genomes are also relatively easy to sequence because compared to other vertebrates, birds have relatively small genomes and a conserved genomic structure. The B10K was initiated in 2010 as the Avian Phylogenomics Project, spearheaded by Goji Zhang and Tom Gilbert from the University of Copenhagen and Eric Jarvis, who is now at Rockefeller University. Back then, they sequenced 45 new genomes, and in addition with some published bird genomes, they had representatives for each of the main lineages of birds, so each of the orders. And they concluded the project with a special issue in Science with 8 papers on the phylogeny, comparative genomics, and genetics of complex traits, in addition to a number of accompanying papers. Based on the large amount of data that was and insight that they were able to get from these few genomes, they decided to start the Bird 10K project. So the Bird 10K project is organized in 4 phases in order to break up the task of sequencing 10,000 bird species into more manageable units. Phase 2 sequences representatives of each of the families. Phase 3 representatives of each genus, and then phase 4 sequences each species. In phase 2, 3 additional PIs joined the board, Carsten Raberg, Gary Graves, and Fu Min Lei. And the consortium also significantly grew. And we are now over 140 consortium members. The B10K has 3 main goals. One is to release the genomes as an incredible resource that will enable a wide range of research. Those draft genomes can be used to do population resequencing of specific species, or they can be used in cross-species comparative analyses to study specific gene families, for example. We're also very interested in generating new phylogenies in each of the phases of the project to get a better understanding of diversification of birds through time, and to uncover the genetic underpinnings of the diversity of bird traits and their modification during evolution. In the following slides, I would like to emphasize a little bit more of how the sampling of the B10K is spanning the bird tree of life. So here I'm showing a synthetic tree from the open tree of life, which with each of the over 10,000 species highlighted as light gray branches. And in blue, I'm highlighting the 48 species that were sequenced in phase 1. In phase 2, a total of 363 species and 218 families were sequenced. And you can see that this tree is much better spanned now. I'm happy to announce that this data will be released in November in a publication in Nature, where we're releasing 267 new genomes and a whole suite of genomic data for 363 species of birds, including a whole genome alignment of all of those genomes. And we're using the data set to illustrate how this dense representation of genomes can really improve resolution for a variety of comparative genomic analyses. As I said, we have started phase 3 as well last year, which will sequence over 2,000 species in each of the genera, and the tree will be quite well spanned. Nonetheless, still 8,000 species will be missing to achieve the overall goal. We have acquired about 50% of the tissues, and we're actively seeking the remainder of about 37% that are still missing. In this phase, we're also changing the sequencing strategy from short read sequencing to single tube long fragment read sequencing, or 10x genomics sequencing, which allows us to get a genome for about $1,000 to $1,500 per genome. We would like to add high-C data if possible, but because we are using museum samples largely, we are often limited in the quality of those genomes to employ this high-C strategy as well. Our project relies heavily on tissue contributors, so scientific collections and museums, and a handful are thanked here individually. We are also always seeking new collaborators who, if you want to contribute tissues of your favorite bird species, or maybe you already have genomes sequenced, or maybe you have ideas for analyses and how you can use those genomes for your questions. So please contact Goji Zhang or any of the other PIs or myself under those emails. With this, I would like to acknowledge the B10K consortium and the leadership and my co-authors, and also the funding that makes the B10K possible. And I'm happy to answer any questions.
All right, thank you, Josephine, for a great presentation and for giving us an update on what's happening with the B10K effort. So we'll now take questions from the audience. Michelle Strickland and I will screen them. So we have one here, Josephine. It says, what annotation strategy was taken for the 360 species in phase 2?
So we used transcriptome data and chicken and zebra finch annotation, so from available genomes, and then annotated using those genomic resources and transcriptomic resources.
Okay, and I'll further ask, or you can add, is transcriptome data from like what kinds of tissue, roughly?
So those were all already published transcriptomes, so we didn't generate new transcriptome data. So those were publicly available transcriptomic data for either closely related species or the same species. And so we basically used everything that was available at the point of about 2 years ago.
Right. Okay. So I'm waiting for some others to come in here, but I'll ask one myself. Is how can somebody get involved if somebody wants to, either contribute a genome or do some analysis? What do they do?
Yes, so the easiest way is just to go onto our website and to message any of the principal investigators or myself, and we will then find the right person for you to talk to. So just email us, email me, Goji Zhang, or Eric, or Tom Gilbert, and we will find a way. We are also looking for tissues. So here we're really interested in obtaining new tissues, and we have a person who is now working full-time in acquiring tissues. So if you contact one of us, we will put you in contact with this tissue, with this person to organize the new tissue samples. And
okay, good. You have a flood more coming in here. Let me just ask a few more. So are you planning to enhance the quality of already assembled genomes as well?
Yes, so we're trying to, if it is possible, and we can get, we have DNA left from previously sequenced samples, we will try to update them with this ST-LFR sequencing, which does give better genome assembly quality. So we are trying to do that wherever possible. And like I said in the presentation, we are also hoping to do some Hi-C, but here really the sample quality is what is mostly restricting those new assemblies. But whenever we can and when we have really good tissue, we are updating assemblies. Okay,
great. Here, are you planning? Well, that was voted up twice. Another one here says, I would like to include transcriptome data in Hi-C in the near future, permit dependent. So just putting a shout out there for Jose Ipirizaga, who said that. And so what are the plans for the B10K project for conservation?
Yes, so I mean, the genomes, we do have a number of specimens that are of endangered or critically endangered species that are being released. So these will be really useful for conservation efforts to do resequencing studies, population genetic studies. We also have a project that is looking at demographic changes. So it's using the genomes to reconstruct ancestral population sizes through time, trying to identify which factors influence population changes through time. So that's a project led by Xiaohong Feng and with Goji Zhang. And
okay, yeah, I'm gonna have to stop it there because we're gonna go over time if we don't. But I would say there are a lot of questions there, Josephine, and people have included their names, so you know who to contact to answer some of these questions, a lot of good ones. So go ahead and we'll get the chat to you later. And thank you very much for that presentation. It sparked a lot of interest, it seems. All right. So next up is Phil Morin from the National Oceanic and Atmospheric Administration. It's going to talk about the Cetacean Genomes Project. So go ahead and take it away, Phil.
I'm Philip Morin from the Southwest Fisheries Science Center in La Jolla, California, and a project coordinator for the Cetacean Genomes Project. Cetaceans include whales, dolphins, and porpoises, and represent not only a unique adaptation of mammals to an entirely aquatic life, but also tremendous diversity of adaptations of interest for research and conservation. For example, the blue whale is the largest animal ever to have lived on this planet. It can reach over 33 meters in length and 190,000 kilograms. The record for the longest annual migration goes to the gray whale, which a single animal has been recorded to travel more than 22,000 kilometers in a single round trip, during which it's fasting. The oldest mammal to date is the bowhead whale, with estimates that bowhead whales can live to over 200 years. There are two records for the Cuvier's beach whale, one for the longest dive ever recorded at nearly 3 kilometers deep. And recently, a single dive was recorded at 3 hours and 42 minutes. The killer whale is the most widely dispersed mammal on the planet. This is considered to be one species, but it ranges through all the oceans of the world. It's found in habitats ranging from the high Arctic and Antarctic in the ice to the tropics. It is adapted to feed on different types of food and live in different types of habitat. So we're learning through genomic analyses across many individuals the diversity of this species that involves a very complex evolutionary history. We're discovering new species of cetaceans on a regular basis. Just last year, the Sato's beach whale was first described. And despite the fact that this whale grows to 6 to 7 meters in length, it's only been documented from dead animals that have been found on the shores of Japan, Russia, and the Aleutian Islands of Alaska. And finally, the reason that I got into cetacean genomics is for conservation. The majority of cetaceans have been impacted through commercial whaling or other impacts on populations throughout the world. And there are current and ongoing and developing threats to them throughout the oceans of the world. I started working with the genome of one species a couple of years ago, and that's the vaquita, which is a very small porpoise that's found only in the northern Gulf of California, and is the most endangered marine mammal. The poster that on this slide to the right was from a couple of years ago when it was estimated that there were fewer than 30 vaquita remaining. Presently, it's estimated that there are fewer than 20 remaining. And we use the genomes to understand something about this rare and poorly known species. We looked at heterozygosity across the genome to both determine how diverse it is, understand its evolutionary history and relationships, and to examine its demographic history to understand how it's persisting at a small population size in this very isolated range. What we learned from the demographic analysis is that the low diversity in the vaquita is due to a very long-term stability as a small population. For over 250,000 years, the vaquita has persisted as a small population and therefore has likely shed a lot of its deleterious mutations and mutational load so that it may be able to recover from the current very small population bottleneck if we can get nets out of the water so that it has a chance. So we started the Cetacean Genomes Project just over a year ago as an international collaboration to establish and coordinate efforts to sequence the genomes, reference quality genomes from all cetaceans. Our primary goals are to collect and organize information on samples available from accessible collections and solicit appropriate samples from the scientific community, and coordinate and disseminate information on the best practices for sample collection and preservation for ultra high quality DNA and RNA samples. We also coordinate available data to avoid redundancy and reduce costs of completing reference quality genomes, and seek funding in some cases for individual or groups of species in coordination with researchers with interests in genomic analyses. The primary coordinators are myself, Andrew Foote in Norway, and Michael McGowan at the Smithsonian Institution in Washington, D.C. Though we coordinate with other researchers around the world and directly collaborate with the Vertebrate Genomes Project and the Darwin Tree of Life Project for sequencing and genome assembly. We also try to facilitate other projects for genomes of cetaceans, including the DNA Zoo, which is producing high-C assemblies of cetacean species. To date, the Cetacean Genomes Project includes 3 completed cetacean reference quality genomes out of the 90 recognized species. These are the vaquita that I just spoke about, the blue whale, and the bottlenose dolphin. We also have 6 more projects in progress at the moment, and these were chosen based on availability of samples and also to represent the diversity across the families of the cetacean phylogeny, and to help us build reference quality genomes that can be used for mapping of genomes from other more closely related species in the different parts of the cetacean phylogeny. Samples are by far our largest issue. We're not able to go out and collect whole animals and obtain samples and cryopreserve them. So we have to rely on a couple of other methods. Primarily, we rely on tissue culture from DART biopsies. DART biopsies still require a tremendous effort to get out into the field to find species, especially rare deep ocean species, obtain the biopsy, preserve it properly, get it back to the laboratory, and culture it. In rare cases, we're also able to get fresh, high-quality tissues from beach cast animals and captive species, where we can obtain tissues and quickly cryopreserve them or establish subcultures from these tissues. I've been working for over 15 years with the frozen zoo collection at the San Diego Zoo Global to try to establish cell lines so that we can do genomic types of research. To date, we've been able to culture 17 cetacean species in the collection. Each of these expands a tiny little biopsy into millions of cells that can then be frozen and then used for ultra high quality DNA and RNA extraction and chromatin mapping protocols. So we plan in the next year to complete 6 or more reference genomes and continue to coordinate collection of appropriate samples so that we can get closer to the goal of sequencing all cetacean species. This won't all be done within the couple of laboratories in the United States and the UK. Rather, it's an international effort, and we are working to coordinate genome sequencing internationally, helping to build capacity and help people communicate, coordinate capacity for sequencing and assembly and expertise to be able to build genome assemblies in countries where the species and researchers are found. More information on our project is available on our website, or you can contact me by email. Thank you for your attention.
Thank you, Phil, for that exciting story on the vaquita genome and the Cetacean Genomes Project. I want to say that I'm actually thrilled for this project, as the previous one we heard from Josephine as well, because cetaceans are vocal learners, one of the few groups of animals that can imitate sounds. And we got some birds in that category as well. So, so great work. So here's the first question from Anshwash Saha. So my question is not directly related to cetaceans. I work, I work on, I'm going to have to pronounce this correctly, see, Eucontoglires phylogeny, how to collaborate with the VGP regarding sequencing facility. So Phil, I'm going to direct that question to you because it's like somebody who's working on a group of species and they want to collaborate with the VGP. And so, you know, how did you do that? How did you come to us? Tell them. You know, we've got to give a brief answer though.
Yeah, I don't do sequencing on large scale myself. And when I heard about the VGP through the Biogenomics conference several years ago, I thought this is a great opportunity to connect with people who have all the expertise in sequencing and genome assembly and leave me to do the science about cetaceans and analyze the genome. So I basically just reached out to yourself, Eric, and Federico and others and said, how can we do this? And you know, it meant getting some money together, but other than that, it was a nice, easy collaboration.
There you go, Anshwa, that's the answer. Yes, we're highly inclusive for the Cetacean project, for the VGP project, for the bird project, and so on. Don't hesitate to reach out. So with 96 species, it's one of the fewer groups that have it, where you can actually maybe even do all the species before the entire project of vertebrates is completed. So how many samples are already in storage?
We have samples for every species that we know of at the moment, but we don't have good quality samples because we can't go out and harvest animals. So the hard part is getting those really good quality samples. And so we've got about 25 of them in cell culture, and probably another 15 to 20 from frozen tissues that were collected from animals that were either euthanized in a captive environment or collected very fresh from an animal that died on the beach. So we're maybe close to halfway there, between 35 and 50% of the way. It's going to take a lot of work to get that last batch.
All right, good. Halfway there for samples, and now we need the money for that half as well, right?
We need the money.
Yes.
Fortunately, I should say that there are more genomes in progress, not to the reference quality level for the long read sequencing. The DNA Zoo is going to probably reach about a third of the species this year. They're doing just the high C and short read sequencing. So we're getting lots of genome resources out there, but we want to get those reference quality genomes. That's going to take the funding and the high quality samples.
All right, by popular vote, 7 so far, we got a question from Dan. Are you going to survey genetic variation from museum samples for some of the near extinct species?
Yes. We have museum samples, we have collections at our facility and other NOAA facilities to do population level studies, and we're doing those as we speak. We're doing those on a number of species. So we're trying to understand a lot more about the phylogeography and population structure of these species.
Okay, another one here from Mark Palmeda. I've got some more votes. Philip, such an amazing presentation. I just have one urgent question. Is anyone doing something right now to save vaquitas?
There are a lot of efforts. The one thing that has to be done is to stop gillnet fishing in the Northern Gulf of California. And that's the focus of everybody's work, because it's essentially all that can be done to keep them from going extinct within the next couple of years. And I can't speak to everything that's being done. There's international efforts to make that happen.
Okay, last question here. I'll make it a combined question from Camila and Joanna Damas. Is how similar or different the chromosome numbers in cetaceans? Anything peculiar about their chromosome evolution?
From what we've seen so far, their genomes are pretty highly conserved. They typically have 21 or 22 chromosomes throughout all of cetaceans. And so we haven't done a lot of comparison yet. We only have the 3 completed reference genomes. And the blue whale was just released yesterday at NCBI. The vaquita's been out for about a year. And I think Tursiops, the bottlenose dolphin, still hasn't been released fully, the newest version. Eric, you could speak to that. But yeah, we haven't done.
It'll be out soon. Yep. Okay, I'm going to have to stop there for the sake of time. Very good. Lots of questions there as well. And thank you, Phil. All right, for our next speaker coming up, Michael Westbury from the University of Copenhagen as well. And we'll be talking about speciation in toothed whales based upon analysis of their genomes. So Michael, go ahead and take it away.
Hi, it is my pleasure to introduce our work where we use published genomes to look into the speciation process within the toothed whale superfamily Delphinoidea. So a little bit of an introduction. So speciation is the divergence of lineages through reproductive isolation. And while there are multiple different modes of speciation, allopatric speciation, sympatric speciation, and parapatric speciation are the ones I will talk about today. So allopatric speciation is thought to be the most common mode of speciation. So this is where a geographical barrier separates individuals within a population, which eventually leads to reproductive isolation between those separated individuals, and through enough time will lead to speciation. Sympatric speciation is where the individuals within the population start to differentiate from other individuals within a population without geographical isolation. This could be through some sort of ecological isolation, and eventually, with enough time, these ecologically diverged individuals will then lead to new species. And finally, parapatric speciation, which is a little bit like a hybrid of both of the other modes, where partial but incomplete physical restrictions to gene flow eventually lead to genetic isolation and speciation. And this is more common in wide-ranging species. So speciation in the marine realm is an interesting example where there is a bit of a paradox where it is thought that because of a lack of geographical barriers and because of the long distance dispersal capabilities of a lot of the species, there can be no allopatric speciation, which is thought to be the more common form of speciation. Which brings us to our study. So this was to look into the marine speciation paradox in cetaceans, specifically because cetaceans are highly mobile, but also have a diverse range of species. So the question is, what was the speciation process within Delphinoidea? Was there allopatric, where there was no post-divergent gene flow, or some sort of sympatric or parapatric speciation process where gene flow did occur after the initial divergence of the lineages? So the question was to look into, was there a presence of gene flow after the initial divergence of lineages? And if there was, how long did this continue for? So a little introduction into Delphinoidea. So it is a superfamily within the toothed whales, and it contains 3 extinct families. This includes Delphinidae, Phocinidae, and Monodontidae. And the phylogenetic relationship of these are the Delphinidae is the sister species to Phocinidae and Monodontidae. So what makes this superfamily extra interesting to study the process of speciation with gene flow is that within each of the 3 families, there are contemporary hybrids that have been presented. So this is different species coming together and forming viable living hybrids. So this makes us question whether there's ongoing gene flow. What is the status of reproductive isolation between all of these different species? And what was the initial process into this speciation? So what we used was only published whole genomic data from representatives from all 3 families, including the outgroup Baiji, or the Yangtze River dolphin. So first thing we did was to look into the sliding window phylogeny. So we broke the genome up into multiple different window sizes, ranging from 50 kbs up until 1 megabase. So what we could see here is the 50 kb windows phylogenetic tree, and these gray lines here represent each individual window, the tree for each individual window, and then the black lines represent the consensus tree. And as you can see, when you compare 50 kbs to 1 kb, there was a lot more of a mess around Delphinidae with this very gray cloud of the individual windows, compared to a lot more differentiated within the larger windows. So this shows us that there is a high level of discordance, at least within Delphinidae, and the more discordance within the shorter windows may lead to the hypothesis that it is older events rather than more recent events that are causing these discordances. However, just looking into phylogenies cannot help us to differentiate whether this is caused by gene flow, these discordances, or whether incomplete lineage sorting played a role. Which brings me to the next analysis, which is the D statistics, or ABABA, which can be used to differentiate between incomplete lineage sorting and gene flow. It uses this 4-taxon approach here, where you can detect gene flow either between H1 and H3 here, or H2 and H3 here. So for this analysis, we looked into every single possible combination that is phylogenetically correct based on that topology I just showed you, the species topology, and out of pretty much all of the comparisons, we found some sort of sign of gene flow. So this shows even not only between families there was gene flow, but also within families. So the evolutionary history of this superfamily was likely very, very complex with a lot of gene flow, which leads to the question of how long did this gene flow continue after the initial divergence? And based on the fact that we see contemporary hybrids, is it still ongoing between some of these different species? Which brings me to the F1 hybrid PSMC, which can be used to date the end of gene flow between 2 different genomes or lineages. And for this, you take the genomes from two different species, pseudo-haploid them, and fuse them together to make one single pseudo-diploid. And you run a PSMC analysis, which shows that when there's a large increase in NE, like you see here, that is when most of the differences between the two pseudo-haploid sequences coalesce, meaning that this is likely the point where gene flow ceased between those two lineages. So the basic summary of the HPSMC results was that despite diverging 15 million years ago, Monodontidae and Phocinidae continued to exchange genetic material for over 10 million years, where it ceased about 6 to 4 million years ago. A similar pattern was also seen when comparing Monodontidae to Delphinidae, where despite 20 million years ago diverging, gene flow continued until about 7 to 4.5 million years ago. And again, a similar pattern is seen between Phocinidae and Delphinidae, where despite diverging 20 million years ago, gene flow only ceased rather recently, somewhere between 8.1 and 5.1 million years ago. And just for your knowledge here, this is every single species comparison, pairwise species comparison. And the take-home message of this is that gene flow here ceased before current. So gene flow has ceased between all of these lineages now. So the results from the HPSMC just showed that continual gene flow has occurred for millions of years after the initial divergence of the lineages, and most significant gene flow events have ceased between all of the lineages we compare. So the conclusions of this is that speciation within Delphinoidea occurred in the presence of gene flow, and it was probably a very long and slow process until complete reproductive isolation between many of the lineages. Which leads to the thought that it was most likely sympatric or maybe even parapatric speciation given the ecology of these whales, and that the contemporary hybrids most likely have occurred at either a very low frequency or are highly negatively selected against, so they're not really contributing to the parental gene pools. And with that, I would like to acknowledge all of the co-authors on the project and also the uploaders of the original genomic data, without whom this project would not have been possible. And thanks for listening.
Thank you, Michael. Very interesting results. Nice to see what you can do once the genomes are sequenced, and amazing that you can actually infer when gene flow stops and starts. And I'm curious, so it looks like 5-10 million years ago, gene flow was stopping in a bunch of these whale species. Is that coincidental? Is there any kind of thing happening in the climate at that time that can explain that result?
Ooh, that is a good question, and I'm not sure what was going on in the climate at that time. It could have just been that 10 million years ago, they kind of, the genetic differentiation occurred so slowly that eventually, as time continued, they eventually just couldn't exchange genetic material anymore or produce viable offspring.
Do you think that this gene flow pattern is, of course, these cetaceans have long-lived lives, but are they a telltale sign of what goes on in speciations generally, or the timing looks different in cetaceans compared to other groups of species?
Well, I only know about it in a few other examples, and it seems to be that in cetaceans, the gene flow does occur or continue for a lot longer, which brings me back to the idea of parapatric speciation with the lack of gene flow. So they can just continually exchange material for a longer period of time, as opposed to terrestrial animals, for example, that can't move as much and are more isolated from one another.
Okay, all right, here's a question from an anonymous question. Are some genes more likely than others to jump between species?
Because we only had the single genomes per species, I wouldn't say I would be confident in looking into that, because it could just be incomplete lineage sorting as opposed to gene flow. But if we had population level data from all of these individuals and you found it in all of the individuals within the population, maybe then I would be able to answer that question a bit more reliably.
Which means we need more genomes, even.
Well, Phil's working on them, so.
Yeah. All right, a question from Saran. Do you have species or subspecies level of genome assemblies for your work? I guess,
So
I guess the question is subspecies. Or
subspecies, I mean, that's also tricky. What do you really call a different subspecies? But I just kind of looked up into GenBank and what was available at the time and just took everything. So there are, for some of the species, there are multiple genomes available that we could have looked into, but for the general purpose of this study, I just took one, aimed for one per genera, apart from the Tersiops where I took two.
All right, can you comment on the role of sexual selection and potential delineation of gene flow? And I think that'll have to be the last question. Go ahead.
Well, all of this study was just looking into the autosomal patterns. So I can't really say anything into sex-related gene flow, but I know there are discordances between mitochondrial genomes and the nuclear phylogeny. So I'm guessing there is some differentiation between sexes and gene flow.
All right. Okay, well, thank you. I want to say to you, the most upvoted question is from Tali. It says, terrific talk.
Oh,
All right.
thank you.
Yes, so you got a lot of good comments out there about your talk. All right, I wish we had time for more questions. You'll see them there in the chat box for you to look at them later. And next, I'd like to introduce Stefan Pauls from the Lowy Center for Translational Biodiversity in Frankfurt, Germany. And it's going to talk about a large-scale genome project that I and others like to hear about that they're spearheading, or genome effort.
Hi, I'm Stefan, and I'm co-speaker of the Lowy Center for Translational Biodiversity Genomics that I'd like to introduce to you today. The Lowy Center or Lowy TBG as we call it, is about making the genomic basis of biological diversity accessible for basic and applied research. So our research is driven by questions in ecology and evolution, and we then try in 4 different fields of research to address these questions using genomic information from a wide variety of organisms across the tree of life. The 4 different research fields are comparative genomics, natural products genomics, genomic biomonitoring, and functional environmental genomics. While comparative genomics is basic research by its general nature, the other 3 research fields are much more applied, and we use the transfer unit to try to get results or potential applications from these research fields into the marketplace. All of our research is based on genome sequencing. And for this, we've established a bioinformatics and laboratory unit that I want to introduce to you first. So our laboratory center is mainly focused on new protocol development. And there's a couple of areas that this really focuses on. It's field-based technologies because as biodiversity researchers, we're often in the field. And also trying to obtain good quality high molecular weight DNA from a biodiverse array of samples. And this is important because as we all know, different organisms lend themselves more or less, better or worse, to high molecular weight DNA extractions. So there's a lot of optimization that our laboratory center does. Then we're also really interested in low input DNA sequencing, especially long read sequencing. And the reason for this is that a lot of the biodiversity that we're trying to sequence and get genomic resources for is really small in nature. And this is an area where we've teamed up with PacBio and the Max Planck Institute in Cologne and have made some relatively good progress. So if you're interested in this, I would suggest that you go to the poster by Clement Schneider and look at the long read de novo sequencing that he's done from a single springtail specimen. Now, with the help of the laboratory center, we've sequenced over 200 different species from across the tree of life, including some areas where genomic resources had previously not been available or hardly any had been available. Now, let me briefly introduce to you the different research fields. Comparative genomics is about comprehending the origin and functional adaptations of diversity. And one example of this is studying how gene flow, current or past, might affect speciation and species boundaries. And this is research by Axel Janke's group looking at giraffes and the African giraffes, the 4 species that are currently recognized. And he wanted to assess whether or not there's gene flow between them and whether or not the level of gene flow that does or might exist actually is too high to allow maintenance of species boundaries. As you can see from the plot here on the left, there's relatively little gene flow, but there is gene flow among all the species, but it's not sufficient to really break down the species boundaries. Another example of comparative genomics is studying genes and genome structure to illuminate ecological diversification. And this is one example where we're doing this in caddisfly silk. Now, if you're interested in caddisfly silk, its ecological functions or its evolution, then I would suggest you go to the poster by Paul Franson, who'll be talking about the evolution and differentiation of silk genes and how that links to different silk uses, or the talk by Jacqueline Heckenhauer, who'll be talking about genome size evolution in caddisflies. In natural products genomics, we study the genomic basis of biologically active substances. And one example of this is a proteotranscriptomic analysis in a predatory ribbonworm. Now, this study was interesting for two main results. One is that the authors found two new putative nemertein toxin classes, so classes of toxins that had never been discovered before. But also, there was a link between the evolution and the ecology of these ribbonworms. So scavenging ribbonworms generally produce defense toxins that they produce with the mucus on the outside of their body. Now, the predatory species apparently have modified these defense toxins that were produced in the mucus into predatory toxins. And these are now not only produced in the mucus on the outside of the skin, but actually also in the glands that can now be injected directly through the stylet into the prey. Another example of natural products genomics is about environmental adaptation and stress tolerance. So colleagues looked at lichens from temperate and Mediterranean climates. And what they found is that there's a certain biosynthetic gene cluster that was always present in the temperate climate, but never in the Mediterranean. If you're interested in this study of environmental adaptation, then please go to the talk by Garima Singh on Thursday at 6. In the field of functional environmental genomics, we're interested in detecting functional changes in individuals, populations, and communities before the communities are subjected to long-term change. If you're interested in looking at, for example, pollution-caused mutation rates or changes in allele frequencies, or also functional changes in communities, then I'd like to recommend that you go by the posters from Halina Bindadoria, Miklos Balint, Peter Decker, or Anna Küchler. In the fourth field, genomic biomonitoring, we use genomic data to inform applied biomonitoring schemes and conservation efforts. And one example of this that goes beyond metabarcoding, for example, is the development of a microfluidic array for rapid species detection and also tracking of individuals. So this is an example where we looked at a community of fish, amphibians, and pathogens. And using the microfluidic array, we can do a semi-quantitative detection from an eDNA water sample. Now, this allows us to look at the interaction and monitor the interaction between an invasive predatory fish in this case and an endangered amphibian species. And we can monitor this at any given time and through time to see how these communities' interactions change. So LeuVe-TBG is carried by 4 partners: Senckenberg Natural History Museum and Research Institute, the Goethe University in Frankfurt, the Justus Liebig University in Gießen, and the Fraunhofer Institute for Molecular Ecology. And we've been lucky to receive funding by the state of Hesse for 4 years so far. We're hoping to get a 3-year extension through the state of Hesse, through which we can further develop our 20 core projects and about 15 to 20 associated projects. But most importantly, our institutions, the funding or the partnering institutions, have committed long-term to biodiversity genomics. And so we've been able to hire 4 new permanent professorships through LeuVe-TBG, and we've been able to establish the bioinformatics and genomics service unit, which are there for the long term. And we don't work in isolation, and we've got partners across the world helping us and collaborating with us on our projects. And if you've got a project that you think might be interesting for collaborations with TBG, then please don't hesitate to contact us. And with that, I'd like to thank you very much for your attention, and especially the organizers for organizing this and allowing us time to present TBG. And I'm happy to take any questions.
Thank you, Stefan. Thank you for telling us some wonderful stories that are happening at your center. I have a quick question. The 200 species which you sequenced, which sounds like with PacBio reads, so have they been assembled, sequenced with something else? And also, could they be, will they contribute to some projects out there, like the Vertebrate Genomes projects and others?
Yeah, thanks, Eric. So the over 200 species that we've done are part of about 1,500 genomes that we've currently sequenced so far, and a lot of them are resequencing samples, and those 200 are a mix of different technologies. So a lot of them are based on hybrid, long read, short reads, but the most recent ones are all basically PacBio. We definitely want to contribute these sequences in the long run to the different initiatives, and we're working together with Earth Biogenome on this and also with ERGA on this to basically develop or to contribute to the larger initiatives. But we're happy to talk about contributing to individual ones.
All right, looking forward to that. How did you decide that the genomic sequences support 4 giraffe species?
Pardon?
Yeah, how did you decide if the genomic sequences support 4 different species?
Okay.
One,
yeah, so this is work from Axel Janke, and I can't give all the details, but this is basically on limited gene flow. So there's, of course, morphological evidence, but the limited gene flow from the genome sequencing analysis based on the multiple specimens indicates that they're good species boundaries.
Right, okay. All right, good. And I'll just say, how did you visualize combating antimicrobial resistance with your approach?
Combating antimicrobial resistance?
Yeah. Yeah, I guess that's something you didn't cover. Okay. All right.
Not sure about that. So antimicrobial resistance is not something that we're focusing on a lot. What we did look at is in the ribbon worm study, if this is what this is referring to, then this is just where the venoms basically change from being defense venoms to predatory venoms that would be able to be produced through the stylet then.
Okay, good. All right, well, we're gonna have to stop there for the sake of time. I'm gonna tell the audience that we're going to continue until we complete the full hour, because after this is a concert. And I don't think it'll take too long to log into that. So well, thank you, Michael. I mean, Stefan, a great story. So last but not least is Sri Raj Kolora. And we're gonna hear about, from Peter Stumont's group, we're gonna hear about extreme longevity in rockfish, and then take your questions afterwards. So go ahead and take it away, Rohit.
Hello everyone, hope you're all safe and doing well. I'm Rohit Kolora, a postdoctoral researcher at the Decorative Biology Department at University of California, Berkeley, close to the San Francisco Bridge. I work with Professor Peter Sudmant on rockfish evolution. I'm really excited to introduce you to our research on rockfish evolution, primarily focusing on the aging phenotype through a comparative genomic approach. Aging is inevitable. This is common across all biological life forms. With increasing age, the proficiency of life forms to function normally generally deteriorates. This is referred to as senescence. As we senesce, the capability of our cells to repair themselves or regenerate decreases. This makes us more vulnerable to diseases at older ages. Aging or gerontology is an active area of research across various grades in the tree of life. Vertebrates, which are known to have originated more than 500 million years ago, exhibit a huge variation in lifespan, more than 2000-fold, as you can see in the case of pygmy gobies, which are known to live less than 2 months, and Greenland sharks, which live more than 400 years. Our model system of interest, the rockfish, there are some species which are reportedly living more than 200 years in the wild. Across vertebrates, hallmarks of aging such as sensory dysregulation, loss of genomic stability with increasing age have been identified. Recently diverged species help study specific phenotypes due to their shared ancestry. Our focus is to study recently diverged rockfish species through an evolutionary perspective, mainly the aging of rockfish. But why rockfish? Fishes are known to make up more than half of the known vertebrate species. The rockfish family, Sebastidae, exhibit the third highest speciation rate within fishes. So the genus Sebastis, generally referred to as the rockfish, are known to contain more than 100 different species, where this is highly species compared to their sister genera, which are represented by less than 10 species. Rockfish are also known to have extreme adaptations to the ecology around them. This is visible to their morphological variation, as you can see over here. But you might wonder what is so interesting about rockfish in terms of aging. This is a plot of the maximum life information in rockfish, and as you can see, the rockfish are known to live from 8 years to 200 years. This is more than 25-fold variation across 100 different species. What more can you ask from a model system to study aging, right? So in order to study the rockfish, we started this rockfish genome project. This is spearheaded by Professor Peter Sudmant. We collected samples from all over the world. The dots on this map represent the sample location, and the species diversity is represented with this heat map. There are at least 3 known hotspots of species diversity: the Californian coast, Alaska, and the Japanese coast. Till date, we sequenced more than 88 different species of rockfish. We also used not just Illumina sequencing, but PacBio and HiC sequencing for at least 7 of the rockfish species. From this data, we aim to study the evolutionary history, the role of selection in driving phenotypic changes, and genome structural evolution in the rockfish. The genome size of the rockfish ranges from 850 to 900 megabase pairs. From the genomic data, we constructed 5 chromosome-level assemblies and more than 70 good quality assemblies. Our N50s of the high quality reference quality genomes is more than 30 MB, and for the good quality genomes, at least 70 of them, we have N50s consistently exceeding 100 KB. This is shown in the figure over here. The x-axis represents the percentage of genome covered. The y-axis represents the genome sequence size at that particular percentage. The N50s and N90s have been highlighted. The fossil evidence suggests that the rockfish were present at least 11 million years ago. This figure shows the ancestral state of rockfish in color gradient. The phenotype over here is the maximum lifespan. This starts from 10 to 200 years. We constructed this phylogenetic tree based on the highly conserved single copy genes. The best model suited to our phylogeny and phenotypic evolution is the early best, which is highly probable due to the adaptive radiation of the rockfish. We also observed repeated patterns of evolution increased lifespan, as you can see in the darker blue shades over here. We estimated the lifespan of the pretty rockfish to be around 60 to 75 years, the maximum lifespan. From the initial genomic data, we also investigated the evolution in genome structure, the changes in gene copies. For this, we correlated the read depth information in windows with the aging phenotype, correcting for phylogeny, of course. We observed some gene candidates which were significantly correlated. These were related to biological processes of immune response, metabolism, transcription factors, and other signaling pathways. One such interesting candidate is the butyrophilin genes, which I've shown over here. In the long-living rockfish, the number of copies of butyrophilin genes is higher compared to the short-living rockfish. But what are butyrophilins? Butyrophilins are members of the immunoglobulin superfamily. These are regulators of immune response and attenuators to tissue-specific inflammatory response. In jawed vertebrates, there are at least 16 known paralogs. Within humans, these are clustered on chromosome 6. There are 7 copies in the MHC1 complex. And whereas in mice, we just see 2 copies. In rockfish, we observed that the butyrophilins are clustered on 2 different chromosomes, chromosome 21 and chromosome 1, shown on the left and right over here. We highlighted the gene density starting from blue to orange. The orange triangles represent the gene directions. The outgroups species is on the extreme left. This is ordered by phylogeny. We observed a significant loss of gene copies in the shorter living rockfish species compared to the long living ones. And we also observed genomic rearrangements or structural variation within this rockfish. We are currently working on other lines of evidence to infer relationships between gene families and aging to ultimately understand the cause for senescence in rockfish. To summarize, we analyzed the origin of the lifespan phenotype in rockfish. We constructed the genomes. We identified gene candidates correlated to aging. These also overlap the known hallmarks of aging. We highlighted one such candidate, the butyrophilins, which have lost the genes in the shorter living rockfish. This is a work in progress, so please stay tuned for more updates from our fabulous work of rockfish evolution. With this, I would like to thank our group leader, Peter Sudman, for effectively leading this project, Gregory Owens, who is currently at UBC Canada, for working on this project together, members of the Sudman Lab for their input and lively discussions, our collaborators for their active support, and UC Berkeley for providing the environment to carry out this awesome science. I'd like to thank the organizers for providing me the opportunity to present our work at this conference. And last but not least, I'd like to thank you all for listening to our story and rockfish evolution. Stay safe and take care.
Thank you, Rohit. Really exciting work on trying to find out what genes are causing fish to live over 100 years old in rockfish. So we only have time for 2 questions. I'll go ahead and ask one. You mentioned some candidates, but have you actually really identified some candidate genes that could be responsible for the longevity?
Yeah, we did identify many candidates, actually more than 200 to be exact. I mean, and we did see enrichments, which I couldn't present here, for immune response genes and genes which are associated with DNA damage repair signaling pathways and hemostasis. Yes, we did identify the genes. We do have those lists.
Nice. Related to that, a comment here from Jose Ituzaga says, very exciting work. Would be interesting to compare with bat longevity. So the genes involved in them, and they also are super immune resistant.
Yeah, we are comparing it with bats and killifish and also human hallmarks. Yes, we are using that data too. Thank you so much.
All right, and one last one here. I mean, you're getting comments like very impressive. Mark, Palmetto, sends you lots of claps. All right, so from Saran, how well were you able to cover telomeric and centromeric regions of the genome? With the idea here being, I think, telomeres involved in longevity.
Yeah, so with the genome assembly, we didn't specifically focus on the telomeres, but we did kind of a KEMA-based approach where we looked at KEMers and looking at longevity, looking at phylogenetic correlations. Long story short, we didn't find any, but this is including all the Illumina genomes too. So we have 80 other genomes in addition to the 7 PacBio genomes.
Yeah, so maybe if you look at the right. So if you look at the long read genomes only, you might have the telomeres there, and then can answer that question.
Yeah, we should follow up on it. Thank you.
Yeah. All right, great, Rohit. Like all the other panelists, you got some more great questions up there. We'll send them to all the speakers later for them to get back to you. I want to thank the entire panel. Really great talks, but great to see what's going on there in the vertebrate world. I'd like to next invite you to the the concert that's going on now, Cosmos Sheldrake. And also to say, if you have any questions for any of the speakers today and you don't have their contact information, you can reach out to me, Eric Jarvis. You can find our contact information on the BGP website or the B10K website. So once again, everybody, thank you. And let's go have some fun for an online party. All right.